Hello everyone! Today I'll be showing you how I completed the alignment practice 2. So the first question was asking to do the pairwise alignment using BLAST for test 1 and test 2. So here are the peptide sequences that we need, and I pasted them into BLAST, but you have to specifically click on protein at the top in order for the BLAST to be done correctly. And then I clicked on BLAST, and it showed me the results of test 2. It showed the score, percent identity, and then I clicked on graphic summary where it showed me the alignment between test 1 and test 2, where test 1 was the query, and then it was the actual alignment itself showing the peptides, and then the dot plot at the end. So after I looked at all of the results from the pairwise alignment, I went on to the second question, which said to use FASTA. I opened up the link, and then it said to use test 1 sequence. You had to limit the query to structure UniProt KB PDB database. So here at the top, it was actually already checkmarked for UniProt KB Swiss Prot isoforms, I believe, but then I unclicked that and went to structures and clicked on the last one. Then I pasted in test 1, and then I submitted. And then it brought me to this page showing all of the results. And in our previous assignments of this module, we learned that test 2 was actually thermophilus GTP cyclohydrolase 1. So that was the result I was looking for in this list, it was the 7th result. And then I took a screenshot of this result and I put it into a different document so I could submit it for the assignment. And then it was showing you that the bit score was 30.7, percent identity 26.1%, and an E value of 1.5. Yes, and then it asked, do you find test 2 as an output when you use BLAST choosing the PDB database? So as an output. So we've also actually done this in a previous assignment. So I went to BLAST P, I pasted in test 1, I specifically selected the protein data bank PDB for the database, and I clicked on BLAST. And it gave me these 7 results, but the 7 results did not contain thermophilus. So that helped me understand that FASTA is more sensitive when compared to BLAST, because FASTA gave us this result while BLAST did not. Then I went on to the third question. Run a PRSS test to check whether there is any statistically significant similarity between the two sequences. So I opened up the new link, and then I followed the directions that were written here. So I pasted in test 1 and test 2. But one important thing was that I had to specifically click on the drop-down menu and change it from accession GI number to FASTA format, because before it was not the correct format. And then you had to pay attention to the note saying shuffle sequence equals submit. So then I clicked on shuffle sequence, and it brought me to the results page. It's up here on the top where I believe the parameters for the search were provided. And then the results or what the scores are below, where the bit score was 30.3 and the E value was 0.28, I believe. Yes, and then it asked a question of what do you think this program does? So I answered this and said, I believe PRSS evaluates the significance of a protein sequence alignment, but specifically looks to see how likely the results obtained can occur by chance. From what I have understood, the lower the alignment occurred due to chance, the more likely the results or alignment is biologically relevant. Then I did question number 4. We had to search up this PMID, but the professor said it was no longer available, so I opened it up in a new slide. and then I answered the other questions that were asked. So here's the paper. I'm just scrolling through it a little bit. And then in order to find the papers that cited this specific paper, I used the PMID, and then I went to PubMed and I searched this query, and it brought me to the paper where I made sure that the title and the authors were the same to ensure it was indeed the same. It should be because of the PMID, but I still did this to check. And then when you scroll to the bottom, it brings you to a cited by section, and it brings many papers that cited this specific paper that we had to read. And if you look at it, each paper is actually about like a different organism, such as Plasmodium falciparum, Rickettsia, E. coli, Cranarchaeota, and then I don't know what organism the last one is, but it is not bacteria, I believe. So for the answers, I pasted down the PMIDs for each of the papers that cited it, and then I answered this question that asked how were they similar and if they're biologically relevant. So reading the assigned paper and reviewing papers that cited it helped me to understand that test 1 and test 2 are not the same protein but are similar because the amino acids that compose the peptide sequences may share some characteristics. In the paper, it stated a new fold known as a T-fold has been found in 4 archetype enzymes that perform different functions. This means a peptide pattern has been found to be conserved in proteins that are distantly related and do not have the same functions. When looking at the list of papers that have cited the assigned paper, each subject was about a different bacterium or organism. What is shared between these different bacteria between these organisms is a high conservation of hydrophobic character that has been seen in the T-fold. The paper also stated this has been found in GTP cyclohydrolase 1 enzyme. As test 2 is a GTP cyclohydrolase 1 enzyme in Thermus thermophilus, the results found from the alignment of test 1 and test 2 are biologically relevant. This is because even though the two proteins do not have the same function, they are still distantly related and are thus biologically relevant. And that's everything. Thank you for watching.